This video is going to focus on the diversification of eukaryotes, which is covered in the Freeman chapter 27 of the 7th edition. This may be called Protists in many other Freeman editions, and it's because this is really a chapter about Protists. And when you're looking at the chapter, you should always use the roadmap as a guide for what you should know in that chapter. And what you want to know about Protists is it really gives you an overall view of the types of diversification that we're going to see in eukaryotes as a whole. And I'll talk about that when we look at the phylogeny. But you want to think about how protists are studied, major themes in diversification of protists, and those major themes include morphology. They also include the development of endosymbiosis and the evolution of multicellularity. And we will see innovations in feeding, movement, and reproduction that we'll see later in other eukaryotes. We'll make sure to note some key lineages of eukaryotes, as well as some key groups in protists themselves. If you look at the phylogeny, one of the things that you will notice about the phylogeny is that there is no synapomorphy. Now, for those of you who are not as familiar with talking about phylogeny, synapomorphy means essentially there's no single trait that ties all of protists together into one group. Yes, you could use traits that are also found in other eukaryotes, but using the nucleus is not a trait that is unique to protists because, of course, animals, etc., also have nuclei. And so there is no single trait that ties this group together. And in some ways, it's considered sort of a trash group because it's really everything that doesn't fit into any other group. And what I think about it as sort of an evolutionary experiment. In terms of evolution, there have been there's variation that already exists in this group, and then some mutation happened, increasing that variation. And that is like an experiment. How did this mutation, which resulted in some sort of phenotypic change that allowed for this protist to do something that maybe other protists couldn't do, and was that successful or not? In some cases, it was very successful, and it led to the evolution of things like plants, and in some cases sort of successful, couple of different species, or maybe a couple of dozen different species, or hundreds of species, but not extreme diversification associated with a really innovative adaptation that allowed for them to occupy new niches. So this is something that you want to think about when you look at it. But we know that eukaryotes are going to give rise to our fungi, to our animals, and to our land plants, which are extremely important groups, at least to most of us, right? Because we're animals. So we care about that. So what you'll see is that what happens is there's a whole lot of species that are all around those species. It's not like they suddenly evolved in one group or separately. What we see is that within a group, fungi and animals evolve, but there's also protists that are in that group and share some similarities with animals. And there are some protists that share similarities with land plants. And there are some protists that even share similarities with fungi. So what do we know about the earliest eukaryotes? Well, they had a lot of the traits that we're familiar with associated with many of our eukaryotes today. They had nucleus, they had an endomembrane system, remember that we talked about in 211. They had mitochondria, they had a cytoskeleton, but no cell wall, and they probably swam around using some sort of flagella, and we have some organisms that still have a flagella that we believe may be similar to the one that we saw in our early protists. This is probably a picture you've seen before many times, but what I want you to think about is, well, life started about 3.6 billion years ago, and we didn't see anything that looked like a eukaryotic cell until about 2 billion years ago. So what was happening for 1.5 billion years? Well, who knows? We have some information about that, but what you want to think about is that it took a while for this all to happen, and part of it is also the increase in oxygen that allowed for a lot of what we see in eukaryotes, and the beginning of evolution of eukaryotes is a change in the atmosphere that was associated with maybe the beginning of this all. So here's the phylogeny again. And so we're going to be looking at phylogenies a lot, but this now one is focusing in on these major groups. And what you want to think about is that you see the major groups that we know today are in there with a lot of the protists. So here we see our land plants, and they're in this group of protists that they call plantae. And here you see our fungi, and they're in the Opisthokonta. So you have both fungi and animals in this group, but they also have the choanoflagellates in there. So what this means is that there wasn't one single evolution from there. There were many multiple evolutionary diversifications associated within protists that became the organisms that we know today. And there are a lot of possible explanations for that. But it's believed that there were probably two single evolutionary trajectories that really brought about what we know as the protist group today. And that includes a theory called endosymbiosis. And endosymbiosis is exactly what it sounds like, endo being inside, and symbiosis meaning living together. And so endosymbiosis is really living together with one organism inside of another. And we know that this has likely happened, and this has happened through some larger organism, some protist-like organism, right? Early eukaryote organism engulfing, single-celled organism engulfing a aerobic bacterium. It's likely that this original single-celled eukaryote was probably doing anaerobic fermentation. And so it's not a very successful cellular respiration technique, right? It's not the process is not particularly efficient compared to what we know as cell respiration today. And so it engulfed this aerobic bacterium, and instead of digesting it and processing it, that aerobic bacterium lived within it and then started to produce or act like what we know as the mitochondria today, the powerhouse of the cell, providing energy for that individual. In turn, this probably could have also happened, and probably did happen, with a cyanobacterium, and this would allow the organism to do photosynthesis as well. So in the plant-like organisms, they probably went through the process where they both engulfed the mitochondria and engulfed the cyanobacterium. So this is considered endosymbiosis theory. Lynn Margulis and others have proposed it. Probably happened about 2 billion years ago. And this allowed for the beginning of a beautiful relationship, as I state here, where we have the eukaryote bacterium providing protection and carbon compounds, and the bacterium oxidizing carbon compounds and supplying the host with ATP. So Lynn Margulis, this is a picture of her here. She died in 2011. She proposed this and wrote a formative paper that was rejected, but people thought she was crazy. Remember, I've talked about crazy ideas. Well, this was one considered a very, very crazy idea, because at this time, remember, we had no genetics. We had no way to sort of look and see where these cells had evolved from. And so she was rejected from about 15 journals. They said her research was crap. And it took some time for it to be accepted, substantiated primarily through genetic evidence. And she was vindicated, but it was a struggle. And she came upon this theory by really looking at how symbiosis is so common in other organisms. So she saw many, many examples of symbiosis and thought, well, maybe this is how it happened. She also noticed that there was a lot of other basic observational evidence of symbiosis, or observational evidence about the similarity between mitochondria and bacteria. They were similar in size. So this is showing you an example of that. They had a double membrane, which is similar to each other. They replicated by fission, so asexual reproduction like bacteria. So they both did that. And when they started to look at the genetics of it, when the dawn of being able to examine sequences, what they found was that in the phylogeny, mitochondria, if they looked at the, remember it has its own, remember we talk about mitochondrial DNA, it has its own DNA, and its own DNA was closest to the proteobacteria group, and more similar to that than anything else. And so that provided more evidence to say that this is very likely to how it happened. So these were, and mitochondria also have their own ribosomes and manufacture their own proteins. So this is all very similar to how bacterium work. This is another example of symbiosis that Lynn Margulis observed. So these are, it's a coral. A coral is an animal, and for those of you to look to 12 know that it's an animal, and it does photosynthesis, which is weird. So how could it do photosynthesis if it's an animal? Well, it engulfs a dinoflagellate, a symbiotic relationship with an algae, and this algae lives inside of its tissues. A picture of this is shown here, and it lives inside of its tissues and allows for it to do photosynthesis. So it is able to engulf this and then basically feed upon these algae, these dinoflagellates, and then these provide photosynthesis and energy for the coral. So there are, though, alternative hypotheses. There's a lot of other work that's been done out there, but at this point, this has been the one that provides the most support, especially if we look at it from a phylogenetic standpoint. And this is that exact example that we talked about. So all living eukaryotes have these mitochondria or evidence that their genomes once had these organelles. And the first eukaryotic cells in the fossil record correlate with the first appearance of rocks formed in an oxygen-rich ocean and atmosphere. So that oxygen was really super important for the beginning of the evolution of eukaryotes. Another aspect that we see in protists that's extremely important is the origin of the nuclear envelope and the endoplasmic reticulum. And it's believed based on, or at least the leading hypothesis is that it's based on folding of the membrane, infoldings of the membrane. So this is just shows you an example of that. So you have your chromosome and your plasma membrane, and then you get these infoldings that start to create additional membrane tissue. Remember that when we're looking at the ER and we're looking at the nucleus, it's all going to be very similar in terms of how the membrane is structured. So the leading hypothesis is this, how it probably happens. And there's a synapomorphy that allows you to look at these distinct groups and how we see in protists that there's different types of nuclear envelopes that are associated with potential different ways that infolding happened. And if something happens frequently, it's likely that this was something that maybe was fairly likely to have evolved many times in different groups that created these monophyletic groups with certain types of nuclear membranes. But there are other explanations for how we may have, or how eukaryotic cells may have gotten a nuclear envelope. And I'm just presenting this. It's not necessarily in your book, but it's important to remember that these are hypotheses at this point. They have some support, but there are other theories for how it might have happened, or hypotheses for how it happened. This is an interesting one that talks about blebs that I thought was interesting, and also something that might be of interest to you. So if you'd like to look up this paper, you may. So what about chloroplasts? How do we get organisms with chloroplasts? Remember, plants have mitochondria, and they also have chloroplasts. It's often confused. Students often think, oh, they have one or the other, but they actually have both. And so there is a lot of support for the fact that there is probably protists that engulf cyanobacteria. So we talk about secondary endosymbiosis when we talk about an organism that had already engulfed their mitochondria, and then they are predatory protist, and it engulfs a photosynthetic protist. And that's when we talk about secondary endosymbiosis. So an organism engulfs a photosynthetic eukaryotic cell, and it retains those chloroplasts. And so that would be an example of secondary endosymbiosis. And in cases of secondary endosymbiosis, you would expect these organisms to have 4 membranes, right? Because they're engulfing something that's engulfing something. So then you would have an additional membrane associated if it was secondary endosymbiosis. And we see that. So this is a phylogenetic analysis. And here you can see that we have primary endosymbiosis here that's happening in our plant group. And then there's secondary endosymbiosis that likely happened in other groups. So this would be why we tend to see in these other groups different pigments that absorb different wavelengths. So if you look at your other photosynthetic organisms in these groups, you'll see that they don't have chlorophyll A and B. They may have chlorophyll B or chlorophyll C, etc. So they're going to be a bit different in terms of not just the fact that they have a different number of membranes, but in addition to that, they also have different pigments. And this is achieved by essentially, remember, one protist engulfing another protist. And so that is a different type of endosymbiosis, and we refer to that as secondary endosymbiosis. So some key points to take away from this is that protists are a paraphyletic group. Remember, they don't have a synapomorphy that unites the whole group, but rather we see that they have an ancestral trait that unites most of the group, but not all. So remember that paraphyletic means a common ancestor, but not all of the descendants, some of the descendants. That the biologists study them. They're very important both medically, ecologically, and evolutionarily. We'll talk about that a little bit more in another video. And they have some key morphological innovations which occurred in protists. That's that nuclear envelope, multicellularity, which we'll talk about a little bit later, array of structures that function to support and protection, which we'll look at when we look at the diversification of protists, and that we see that mitochondria and chloroplasts likely rose in this group through endosymbiosis or secondary endosymbiosis. And that's a wrap. That's what we want to make sure to know for our protists. We'll delve into some other topics about protists in other videos. Thank you.